When it comes to metabolism, things are messy, but they're logical. And so rather than trying to memorize pathways and that sort of thing, let's focus on the logic behind these pathways and the logic of metabolism, the logic of how we have all of these interconnecting pathways, all of this crazy stuff going on. But really, at its heart, we can focus on things like the chemical logic. Why are things happening in this order? The thermodynamic logic. How can we use favorable reactions to drive unfavorable reactions? How can we skew the concentrations of things so that we're always going to have a drive to go one direction or the other, or so that we can change direction so that we can use the same enzymes to go in multiple directions? How can we do that? Basically, we can do that in a variety of different ways. We can regulate the concentrations by doing things like compartmentalization, by using things happening in the mitochondria or things happening in the cytoplasm, things happening in whole different organs and cell types, where we can regulate the concentrations of these things differently to control the direction of a pathway. Within the pathway, concentrations can only get you so far, especially if you have a reaction with a really big delta G naught. And so for those, what you want to do is basically regulate the enzymes. We'll see instances where we have, say, the liver has enzymes that other organs don't have. This is why we can do gluconeogenesis. We can make glucose in our liver, but not in other tissues, because the liver has the enzymes that we need in order to do this, but those other tissues don't have that. We can also basically just kind of regulate the availability of those enzymes to actually be where they need to be. So in addition to having them in different cell types, we can have them in the cell type kind of be sequestered, maybe in internal like vesicles, and then only put them on the surface when we're right, when we get the signal. So hormones are going to be a really big thing too. And then when we have the enzymes, we can regulate their activity. So we can regulate enzymes on a variety of time scales. So one, like we talked about before, like that would be slow, would be kind of like transcriptional control. Like, do we actually have the enzyme present? A bit faster is if we've got it present, we've got it made, but it's kind of being sequestered. If we want to really quickly change the activity of an enzyme, we can do this in a couple of ways. One is by covalent regulation. This is typically done by phosphorylation, where we can say have a phosphorylation cascade set off by a signaling pathway that's going to then regulate the activity of these enzymes and therefore regulate the pathways. We'll also see how we can have allosteric regulation. There, instead of having a covalent modification to our protein, instead of having something like a phosphorylation, we're basically just having our enzyme bind to a small molecule. And when our enzyme binds to the small molecule, we can get an allosteric change. The protein is going to basically undergo a shapeshift that's going to make it either active or inactive. And all of this can regulate what pathways are happening when, which direction different intermediates are being siphoned off into. So remember, we basically call everything an intermediate because even if it looks like it's at the start of a of a pathway, or is at the end of a pathway, really we don't have pathways. We have this giant complex subway system where things are happening. Things are going siphoned off into one direction or the other. But if we're looking at one of these giant big maps, it can get really, really overwhelming. And so we like to have some sort of like simplified logic. We can think about things in terms of pathways rather than thinking about them in terms of this giant web. But we need to always remember that we are in the context of this giant web. So although we're going to talk about like cellular respiration as being, okay, we take glucose, we take our blood sugar, and we're going to break it down. We're going to go through glycolysis, turn it into pyruvate. We're going to go through the citric acid cycle, that Krebs cycle. We're going to basically break it down even further, get a bunch of electrons out of it, pass those through NADH and FADH2 into our mitochondria, where we can then cash in those electrons through oxidative phosphorylation to get ATP. And it seems like it's this nice, pretty pathway where things go in one direction. But even if we think about it as a pathway, that pathway can go in both in two directions. Not exactly the same, but basically we can think about glycolysis, the breakdown of sugar to get to this pyruvate step. We can think of this pathway as we can kind of reverse a lot of the steps. In fact, we can reverse like 7 of the 10 steps. And when we do this, we can make glucose through gluconeogenesis. But there's also going to be steps that we can't easily reverse. And these are going to be sites of key enzymatic control. And all of this, though, is happening in the context of all those other pathways. So not only do we have glucose and glycolysis and gluconeogenesis, but we also we'll consider things like, well, what about the pentose phosphate pathway? It's an alternative fate for glucose. How do we decide what goes there? And later in the course, we'll look at things like, what if we want to go make or break down fats or things like this? So remember that all of this is interconnected, and let's try to focus on the logic. Let's try to use this glucose versus glycolysis as kind of, although we're talking about it kind of like it's an either or, this is just kind of like to give us a little example so that we don't get too overwhelmed by the giant picture. But remember that we're in the context of the giant picture, and things are often going to be more complicated than we're going to make them out to be. That being said, let's try to simplify things down and think about this glycolysis as a pathway and gluconeogenesis as a pathway that's going kind of the opposite direction, but it's going to take some detours. So just like when we were talking about kind of thermodynamics and metabolic logic, the logic of kind of pathways and things, we talked about how there would be steps that would be close to equilibrium, and then there would be steps that would be far from equilibrium. We talked also about how there was this G naught, or the delta G naught. Basically, we said we wanted a negative delta G in order for a reaction to be favorable in the forward direction. Now, a delta G naught is basically saying it's kind of inherent. A negative delta G naught would say it was inherently favorable. But then we talked about how the actual favorability was determined not only by that delta G naught, but also by the concentrations, the amount of your products and the amount of your reactants. We talked about how even if you had a really inherently favorable reaction that had a large negative delta G, that if you had enough of your products, you would actually drive things backwards. But the bigger that delta G is, the more skewed your concentrations have to be in order to do that. And even if you had your concentrations really skewed, they wouldn't be that way for long because you'd be driven towards equilibrium. And as you drive towards equilibrium, you lose that drive. So that's why we keep a steady state rather than getting things to equilibrium. We maintain fairly constant levels of things by replenishing what we're taking out. And then we can take out things in order to drive pathways forward as well. So the actual G, the actual free energy change, is going to be different than the G naught, sometimes dramatically so. That being said, if we look at the G inside of our cells, we can see in the glycolysis for the different reactions of the glycolysis. So basically, this is going to be saying in erythrocytes, so in red blood cells, they went and they calculated the actual delta G for each of the steps of glycolysis. And we will review in a second the steps of glycolysis and the logic behind it that we got practice with our activities in class. But basically, we have these 3 steps. This first step where we have glucose to glucose 6-phosphate. This third step where we have fructose 6-phosphate to fructose 1,6-bisphosphate. And we have this phosphoenolpyruvate to pyruvate and ATP. And these 3 steps are going to have these large negative delta Gs, which means that if we were to try to go in the reverse for these reactions, we would need to have concentrations so skewed that basically it wouldn't be possible in the context of our cells. We call these metabolically irreversible. Basically, we can't reverse them just by changing little bits of concentration wise. We need to actually control the enzymes that do this. These 3 steps, basically, so we have this hexokinase step, we have this phosphofructokinase step, And we have this pyruvate kinase step. These are going to be the 3 steps that have that really big negative delta G. So those are going to be the steps that if we want to do gluconeogenesis, if we want to make glucose, those are the steps that we're going to have to go around. Those are the steps that we're going to have to bypass using different enzymes. We're going to see how we can use different enzymes and regulate them reciprocally. So for example, we have the first reaction of glycolysis is carried out by hexokinase. And if in gluconeogenesis, the kind of opposite reaction is going to be carried out by glucose-6-phosphatase. And these can be regulated reciprocally so that when we turn off hexokinase, we turn on glucose-6-phosphatase and things like this. Similarly to fructose, phosphofructokinase 1, which is going to basically take fructose 6-phosphate and phosphorylate it again to give us fructose 1,6-bisphosphate. In the opposite direction, instead of doing things, just taking this enzyme and going reverse, we're going to actually just use a different enzyme, fructose 1,6-bisphosphatase 1, or FBPase. Now, it turns out that this enzyme is actually going to be super duper highly regulated because this is going to be kind of like our first committed step of glycolysis, because once after this glucose 6-phosphate step, after we phosphorylate glucose and kind of like trap it in the cells, well, then it can be used for various things, including like being used for the pentose phosphate pathway, being linked up to form glycogen and things like this. But once we go to this step, this is kind of going to be, we're committed to glycolysis. And so we're going to want to heavily regulate this step. We'll see how we can regulate these enzymes in various ways. Sometimes we're going to regulate them by using like products that are later down the line to kind of like inhibit yourself with the thing that you make so that you don't build up too much. Like you're like, okay, we've made enough now. Our supply is exceeding our demands. We can also respond to our demands. So we can have allosteric regulation by things like ATP and ADP and AMP so that we can respond to energy levels. So we're going to see how all of this is going to come into play. And then finally, the last step that we're going to need to overcome is going to be this step from phosphoenolpyruvate to pyruvate in the glycolysis direction, and that's carried out by pyruvate kinase. To go in the reverse, we actually have to use 2 enzymes. So we're going to have one extra enzyme in our gluconeogenesis step because we're going to have to use pyruvate carboxylase to go from pyruvate to oxaloacetate, and then PEP carboxykinase in order to go from oxaloacetate to phosphoenolpyruvate. So we have to kind of take 2 steps because that reaction was super duper favorable. And so we'll talk more about this when we get there. Let's start by thinking about the logic of the glycolysis pathway as a pathway, though, and kind of review that chemical logic. So why are things happening in this manner? Like, why do we move things, put a phosphate here? Why do we shift things around? It's not just random. It has to do with what makes it then chemically favorable to do the next step. So in glycolysis overall, we can kind of think about it as being split into 2 phases. An investment phase where we have to put into ATP, and a payoff phase where we're basically going to get for ATP as well as to NADH, which remember can then go into the mitochondria, go through that electron transport chain and give us ATP. And ultimately, that's where the majority of our energy is going to be coming from, is actually from that oxidative phosphorylation, assuming we have oxygen around. But first, we have to put in that investment. And so that conversion of glucose to pyruvate overall is going to be exergonic. So it's going to let off energy. And so basically for any pathway to work, it has to be exergonic. But that doesn't mean that every step is going to be super duper favorable. Instead, basically, the formation of ATP from ADP in organic phosphate is endergonic. But yet we're going to do that in our pathway. We're going to make ATP, and this is not going to be favorable, but because we couple it to favorable things, we make it favorable. And so this is what we talked about before, this idea of coupling of reactions. So you can see that here we kind of like split things in two. So when we generate this ATP, we're actually generating two ATPs. So one ATP here and one ATP here, and then we're generating 4 ATPs overall because we split that in two. So we do each of this 2 times, but then we use 2 ATP. And so that's why we end up with a net gain of 2 ATP and 2 NADH. By coupling all of this, basically what we're able to end up with is basically our standard free energy change for the overall reaction is going to be minus 85 kilojoules per mole. So the pathway makes sense. We're able to use this super favorable reaction, to power a non-favorable reaction and overall still have a favorable pathway. So let's talk a little bit more about the pathway and the enzymes involved in their regulation. The first step is going to be taking glucose and phosphorylating it. This is just a simple kinase reaction, except that this kinase is going to be regulated, as we'll talk about. But this is hexokinase. It's also in our liver, we have a form that's called glucokinase, and we'll talk more about that later. But basically what this is doing is it's trapping the glucose inside of our cells. Remember how our membrane is going to be hydrophobic, and so it's not going to just let glucose, it's not just going to want to let a phosphorylated glucose through. And so what we're going to have to do is we're going to phosphorylate it and trap it inside of our cells. Now, we wouldn't want this reaction to be easily reversible because then we would kind of just be letting that glucose flow back out of the cells after we trapped it. But we want our liver to be able to release the glucose that it makes to give it to other cells, because remember, gluconeogenesis is happening in our liver, but other cells are going to need that glucose. And so basically we have this, this other enzyme, glucose-6-phosphatase, that's basically going to hydrolyze that phosphate. It's going to basically use water to break it off. Instead of trying to put a phosphate group back onto ADP, from the glucose-6-phosphate, it's just going to cut that phosphate off. And so this is glucose-6-phosphatase is the enzyme that's going to do this. And it's in our liver, in like a special place in our liver, so that basically it's not going to let the glucose out if you don't want to. So that's going to be highly regulated. And so this is kind of like our first regulated step, although we'll also talk about how just getting the glucose into cells can also be regulated. But this is carried out by hexokinase, and it's going to put this glucose on the 6th position. Sorry, it's going to put this phosphate group on the 6th position. And note that when the kinase is transferring a phosphate, it's really transferring a phosphoryl group. So this oxygen here is the one that came from the glucose. And then this phosphoryl group, so the phosphorus and 3 oxygen groups, not with 4 oxygen groups, that's what's actually going to be transferred. So this is not an oxidation, but this is a phosphorylation, and it's carried out by hexokinase. And we can see that it has a very large negative delta G. So this would not be easily to reverse, which is why we're going to use this different enzyme in gluconeogenesis. Okay, what about the next step? Well, here we just have an isomerization reaction. We're not changing the number or the type of atoms. We're just shifting things around. So we're basically moving the position of this carbonyl, and this is going to free up this end hydroxyl group so that we can phosphorylate it. And so the enzyme that's going to do this is phosphohexosisomerase, sometimes called phosphoglucosisomerase. So you'll see it listed, you'll see it called both of these things. You can see that it's got a pretty neutral delta G, so this is going to be easily reversible depending on things like the concentrations. And we'll see later how this F6P can also be changed into further phosphorylated to give us F26BP, which is going to be an allosteric modulator. But for now, just focus on the fact that we're basically taking this glucose-6-phosphate and making fructose-6-phosphate, which just involves shifting of this carbonyl group. You might be wondering, why the heck are we doing that? Well, it turns out that doing that, we basically free up this oxygen so that we can phosphorylate it and then have this molecule we can have this molecule be cleaved in half and have a phosphate group on each end. And by moving the position of the carbonyl, well, then it's going to set it up so that when we have a reaction later, it's going to basically cut the molecule in half rather than cutting it in uneven parts. Our next step is going to be phosphorylating that hydroxyl group that we just freed up. And so this is going to be that super duper regulated step. This step of phosphofructokinase, or PFK1. And so this is going to be highly regulated because it's going to be that kind of committed step of glycolysis. And so we'll talk much more about that later. But basically here, you're sticking a phosphate group on the other end. You're sticking a phosphate group on this one position. And you're taking that phosphate group from ATP. And so this is being carried out by a kinase, PFK1. It has a very large negative delta G, minus 22.2 kilojoules per mole in erythrocytes. And so this is why we're going to need to go take a different path if we want to reverse it in the case of gluconeogenesis. And we'll talk about that in a minute. But continuing on our story of glycolysis, the next thing that we're going to do is we're basically going to split that molecule in two. So in this step, basically, the purpose of phosphorylating the second end was so that we could split it in two and have two parts that had the phosphate groups, and that these parts were basically interchangeable. And so we'll see in a minute how they can interchange, but first we need to split it in two, and that's going to be this enzyme called fructose bisphosphate aldolase, most commonly just called aldolase. What it's going to do is it's going to take that fructose 1,6-bisphosphate and it's going to cut it in two. This is going to give you two molecules, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. This is going to have a pretty neutral delta G, which means that this is going to be we can go in either direction. We can use this step in both glycolysis and gluconeogenesis. Once we get these two products, well, now what actually happens is that they're going to turn out being basically the same thing, because we're going to have this enzyme, triose phosphate isomerase. Basically, you're going to go from DHAP to glyceraldehyde 3-phosphate. This reaction, although it's kind of like really easily reversible, what's going to happen is that then you're going to use these favorable steps that are going to pull glyceraldehyde 3-phosphate through the pathway. And so you start out with having both these dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. You start with equal amounts of these, but then as you pull glyceraldehyde 3-phosphate away, well, now you're basically going to drive the concentration of that down, which is going to pull the dihydroxyacetone phosphate towards that good old Le Chatelier's, and that's going to make it so that we can pull this reaction forward and just work with this glyceraldehyde 3-phosphate. Note that once you split it in two, you can't really tell what came from what. And so you have these two equal parts, but now everything in this kind of like payoff phase is going to be doubled. And so everything that happens down here, we're going to double it. So that's why we get like 2 NADH instead of just 1, because all of this is happening twice because we split the 6-carbon molecule into 2 identical 3-carbon ones. Well, not identical at first, but then identical. Now what we want to do is we're going to enter that payoff phase. We're finally going to start making energy money, but first we get an IOU in the form of NADH. And we pick up a free phosphate. Okay, so when we see NADH, what we should be thinking is something's going to be getting oxidized because we're reducing NAD plus in order to form NADH. And in this case, we're also going to be phosphorylating. So we have oxidation and phosphorylation. We have an oxidative phosphorylation. And in fact, this is so you can see the reason why I was saying before that a kinase was going to add a phosphoryl group was because here we're actually adding a whole phosphate group. So we're adding 4 oxygens. So we are oxidating this, and we're going to see that that's an oxidative phosphorylation. Now, typically when we think of oxidative phosphorylation, we're thinking about that thing that happens in the mitochondria where we're passing off all those electrons. And that is oxidative phosphorylation, but there you have the oxidation and the phosphorylation separated, whereas here it's happening at the same time. This is happening into this molecule glyceraldehyde 3-phosphate. We're making 1,3-bisphosphoglycerate, and we're doing this using this enzyme glyceraldehyde 3-phosphate dehydrogenase. So as the enzyme name suggests, this is going to be an oxidoreductase enzyme. And this is consistent with the idea that we're adding a phosphate group and we are oxidizing in the process. So this is going to have a neutral delta G. So again, we can use this in either of these directions. Next up, what we're going to do is basically, we're going to take off the phosphate that we just added. Now you might be like, why the heck did we do that? We just took this phosphate group that was freely floating around and we added it onto glyceraldehyde 3-phosphate, and now we're taking it back. Well, we're not just cutting it off. Instead, we're going to be adding it onto ADP to give ATP. So why could we do this? Well, it turns out that we're basically adding we're making a more energetically, like high energy phosphate bond than we would have with like ATP. And so we're able to add this phosphate group onto ADP in order to get our 3-phosphoglycerate. Why we can do this is because basically what we have is we have this really high energy phosphate bond where we have this acyl phosphate. So this is an acyl group where you have a carbonyl next to basically just like a carbonyl attached to something. And here that something it's attached to is a phosphate. So this is an acyl phosphate group, which is going to be high energy. And so breaking it off is going to give us a lot of oomph, and it's going to be favorable. So we're going to be able to then take that phosphate group off of the 1 position, leaving us with 3-phosphoglycerate. So we went from 1,3-phosphoglycerate to 3-phosphoglycerate. And of course, our ATP that we stuck the phosphate group off of 1,3-bisphosphoglycerate onto ADP to give us our ATP. And remember, that's happening twice because we have this whole second part happening twice. Now what we're going to do is we still have that 1 phosphate group, so we still have that ability to make 1 more ATP, but right now it's not in a good place to do so. It's not a high enough energy bond in order to make ATP. But we can set things up to be powerful enough by using phosphoglycerate mutase. Basically what we're going to do is we're going to shift the position of the phosphate over to the middle. And so you might think, okay, well, what's the big deal about sticking it in the middle? The big deal is basically what we're going to end up with is we're going to be setting ourselves up to form a very, very favorable interaction. So this one's not that exciting. We have this really, really neutral delta G. But basically, we're able to take this, make 3-phosphoglycerate, move the phosphate group to the 2 position to give us 2-phosphoglycerate. And now we're going to set ourselves up for the big payoff. So basically, next step we have is basically now that we've moved things like this enzyme enolase, what it can do is it can do an elimination reaction to remove water. When you remove water, well, now what you formed is this enol. So hopefully you remember back to your OChem where you had an enol was going to be like an alkene, so a double bond between 2 carbons next to an alcohol group. And so enol, you can get this kind of like tautomerization where you switch between an enol form and a keto form. So like a ketone, if you think about like moving these electrons over to here, and then just pushing things up and moving over a proton, basically you can switch between these 2 forms. This keto form is going to be much more favored than the enol form. So if we just take like an enol and we let it go, it would like really want to go to the keto form. But what we're able to do, what enolase is able to do is it's basically going to trap it in this by that phosphate group is going to allow it to kind of trap in this enol form. And when you are trapped in this enol form, basically what's going to then happen is it's going to want to go back to the keto form. And so it's going to really, really, really want to go back to the keto form, so much so that we're going to get this negative 16.7 kilojoules per mole of energy if we let it do so. And so we're going to let it release that phosphate group, transfer it to ADP to give us ATP. This is going to then give us this energy that's kind of like the Big Bang at the end of our pathway. And this is going to give us another ATP, which again, we're going to have all of this happen twice. So that's why we get 2 ATP here, 2 ATP here, but we use 2 ATP here. And so basically then we're going to get a net of 2 ATP and 2 NADH. And we get 2 pyruvate, which can go in the citric acid cycle and get further oxidized. But that's only going to happen if there's oxygen around, because that's going to require the oxygen to be our terminal acceptor. But if oxygen isn't around, then we're going to run into trouble, because one of the things about the citric acid cycle was that it was also going to regenerate our NAD+. And so if we don't have NAD+, well, then this whole pathway isn't going to work, because we needed that NAD+ here in order to do this step. And so if we don't have NAD+, well, now we're in trouble. And so we need alternative ways to generate NAD+ if we don't have oxygen around. And this is what we get with like fermentation. And so that's going to regenerate NAD+ without oxygen so that glycolysis can continue anaerobically. So in some yeast and plants and microbes, they're basically going to be able to take things to ethanol. But we're going to take those pyruvates and change it to lactates. So this is done by lactate dehydrogenase, and that's an alternative way to generate NAD+. Another kind of alternative way to generate NAD+ is to basically make glucose, because if we do the reverse pathway, well, then we're going to basically generate 2 NAD+. But this typically isn't like the main reason why we would do gluconeogenesis, but we do get NAD+ out of it. So let's talk about when we would want to do gluconeogenesis. Basically, you would want to do gluconeogenesis when you want glucose. So when you've got plenty of energy, but not much glucose. And when would you want to do glycolysis? When you've got lots of glucose, but not enough energy. And so later we'll see how these are kind of reciprocally regulated by things like ATP levels, ADP and AMP levels, NAD+ and NADH levels. And so we'll see that the enzymes responsible for these kind of bypasses, those steps that we can't easily reverse, are going to be reciprocally regulated largely. So let's introduce these enzymes, these bypass steps, and then we'll talk about their regulation and more. As we mentioned at the beginning, you basically have these 3 steps with these large negative delta Gs that we need to get back the other direction. And so when we're doing this in gluconeogenesis, we're basically going to have to introduce different enzymes. We're going to have 4 enzymes, because we needed 2 enzymes to basically reverse this last step. So this last step, remember, that was in glycolysis. We are going from phosphoenolpyruvate to pyruvate, and that was done by our pyruvate kinase. It was going to be really, really favorable because we have that whole enol keto tautomerization thingamabob. And so we were able to put a phosphate group onto ADP to give us ATP. And that was carried out by pyruvate kinase. To go in the other direction, basically, we're going to need 2 enzymes in a kind of convoluted way. Basically, we're going to take pyruvate, we're going to change it to oxaloacetate through pyruvate carboxylase, and then we're basically going to take that oxaloacetate and use PEP carboxykinase in order to phosphorylate it to give us phosphoenolpyruvate. So we end up having to actually pay for each pyruvate, we're going to have to pay an ATP and a GTP. You need to put more energy in for gluconeogenesis than we get from glycolysis. A slight complication to this is that if we want to go from pyruvate to phosphoenolpyruvate, remember we have those 2 steps. The pyruvate carboxylase converts it into oxaloacetate, and then the PEP carboxykinase converts that into phosphoenolpyruvate, our PEP. The complication comes from the fact that pyruvate carboxylase is located in the mitochondrial matrix, but the oxaloacetate that it generates can't get out of the mitochondrial matrix. So when we're talking about glycolysis, that was all happening in the cytoplasm or the cytosol, basically the general inside of our cells. But then a lot of our stuff is going to happen in our mitochondria. And part of this is that whole compartmentalization thing I was talking about, where basically we can control the concentrations of things to have different concentrations in different parts of the cell, so that we can have conditions that are favorable for one thing inside of the mitochondria, say, but conditions that are favorable for something else outside of the mitochondria. The mitochondria, the reason why we're able to kind of have different concentrations in there is because we've got membranes that are going to be selective. And so these membranes aren't going to let through everything. Most things can't freely go through. And even those things that can go through, they have to go through special transporters, all those different types of transport that we talked about. The mitochondria, it has two membranes, and the inner one especially is going to be very, very picky. And it's going to let through pyruvate, but it won't let out, and it'll change pyruvate into oxaloacetate, but it won't let oxaloacetate through. Therefore, if we want to continue further on in the gluconeogenesis, we need to convert the oxaloacetate into a form that can be taken out. There are a couple of options to do this, depending on what our fuel source is. One option is to use a mitochondrial PEP carboxykinase, that is to do the second step inside of the matrix. A second option is to use a cytosolic PEP carboxykinase, so do the conversion outside of the matrix in the cytoplasm. If we want to do it that second way, however, where we're doing this second step in the cytoplasm, we need a way to generate oxaloacetate in the cytoplasm from the oxaloacetate that we had in the matrix. One way that we can do this is by converting the oxaloacetate to malate, taking the malate out, and then converting the malate to oxaloacetate. So what we're doing in this case is we're going to be reducing oxaloacetate to give us malate, and then we're going to, in the cytoplasm, be oxidizing malate to give us oxaloacetate. So what's going on in these situations? Well, we can see that in the situation over on the left, we're taking an equivalent of NADH, and we're basically transporting it out of the matrix. In the case of the second case, we're generating this NADH in the cytoplasm, but we're not taking any NADH out of our matrix. It makes sense to do this pathway on the right if we have lactate as our original fuel source, because we're already going to be generating this NADH in this conversion of lactate to pyruvate. However, if our pyruvate is coming from other fuel sources, such as directly from glycolysis, we're not already generating that NADH out here in this step. And so it makes more sense that we're then going to transport the NADH out of the matrix and into the cytoplasm. Because remember, for gluconeogenesis, we want NADH, whereas for glycolysis, we don't want NADH. And so gluconeogenesis is kind of using NADH. And so we want to be taking NADH out into the cytoplasm where it can be used. In the case of our lactate as a fuel source, we're already doing that in the cytoplasm. In the case of our other pathways, however, we're not. And so if we want to get more NADH into the cytoplasm for further use, this oxaloacetate to malate pathway is going to give us a way to do that. For now, I don't need you to worry about which pathway is going to happen when. Just know the net effect that we have these two reactions that are needed in order to convert pyruvate back to phosphoenolpyruvate, bypassing that last step of the pyruvate kinase step of our glycolysis. So long pathway, but we get there. So once we're there, we've got our phosphoenolpyruvate. Now we can basically, all these steps were easily reversible. So we can go back all the way up to here, but now we get to that fructose 1,6-bisphosphate, and we need to get to fructose 6-phosphate. So basically all we need to do is we need to chop off the phosphate group. But we're not going to use the same enzyme as we used before. We're not going to use that phosphofructokinase. We're not going to try to take that phosphate group off of the 6th position and stick it, or of the 1 position and stick it onto ADP. Instead, we're basically just going to cut that phosphate group off. So we're not going to get an ATP out of this. Instead, we're just going to cut that phosphate group off. And we're going to need a different enzyme to do this. We're going to need this fructose 1,6-bisphosphatase. We'll talk more about how this is going to be highly regulated, because remember, that was that committed step of glycolysis, and we can regulate this step. We can regulate whether glycolysis goes forward or whether we take things in the reverse direction. And then finally, once we get back past that step, when we have this fructose 6-phosphate, we can easily interchange that to glucose 6-phosphate. That was just one of our isomerization reactions. Very easily reversible, but now we have this glucose 6-phosphate and it's trapped inside the cells. Now our gluconeogenesis is happening in the liver, and the liver's job is kind of like, well, one of its jobs is to supply glucose to the rest of your body. And if we said that the glucose, the reason why we phosphorylated the glucose was to trap it inside of our cells, and now we're saying that basically it's phosphorylated, it's not getting out of the liver until the liver dephosphorylates it. And again, in this case, we're just going to use a phosphatase, which is a hydrolase that's going to basically use water to cleave off the phosphate group to give us glucose, which can then get taken out of the liver. It can get into the blood, and other tissues can use it.